Revision. Megagametogenesis. The development of the embryo sac or female gametophyte from the haploid functional megaspore situated inside the nucellus of the ovule. The female gametophyte of angiosperms may be classified into three different types, namely monosporic, bisporic, or tetrasporic. Coming to the first one, that is monosporic type. Here, only one functional megaspore nuclei is involved in the development of the embryo sac, and this is also known as polygonum type. In this monosporic type, the first division of the functional megaspore gives rise to two nuclei. After the first division, the two daughter nuclei move apart to the opposite poles, the primary micropylar and the primary cellagil region. Most of the cytoplasm is aggregated around them and the rest form a thin peripheral layer, the center being occupied by a big vacuum. The second division produces one pair of nuclei at micropylar end and one pair at the cellagil end. And again, this undergoes one more division, resulting in two groups of four nuclei at the opposite pole of the embryo sac. The micropylar quartet differentiates into three-celled egg apparatus and the upper polar nuclei and the cellagil quartet into a group of three antipodal cells and the lower polar nuclei. The two polar nuclei fuse to give rise to a secondary nucleus. This type of embryo sac is most common and therefore it is designated as normal type. Coming to the second one, that is bisporic. The bisporic type of embryo sac are typically 8 nucleate, also known as ileum type. Here, the megaspore mother cell divides to form two dyad cells, of which the upper is smaller and soon degenerate. The nucleus of the lower divides to form two, and this megaspore with two nuclei enters the formation of embryo sac. Here, the development of the embryo sac only need two further divisions to give rise to 8 nucleate stage. The mitotic division produce two pairs of nuclei at the micropylar end and one at the cellagil. And the second results in two groups of four nuclei lying at the opposite poles of elongated embryo sac. The micropylar quartet differentiates into three-celled egg apparatus and the lower polar nucleus, and the cellagil quartet into three antipodal cells and a lower polar nuclei. Coming to the tetrasporic type. Here, this megaspore mother cell undergoes first division to form two nucleate stage without the formation of cell wall. And at the end of second division, the megaspore is 4 nucleate. Next, there is 1+3 arrangement of the megaspore nuclei so that the three cellagil nuclei come to lie very close to each other. During the next stage, the micropylar nucleus divides normally, but the three cellagil ones spindle fuse to form a single common spindle, so that at the close of division there are two haploid nuclei at the micropylar region. Then one more division occurs, resulting in 8 nucleate, of which the 4 cellagil nuclei are triploid and 4 micropylar are haploid. The mature embryo sac, therefore, consists of 3 haploid cells, that is, the egg and 2 synergids, and 3 triploid cells, the antipodals, and a tetraploid secondary nucleus formed by the fusion of 2 polar nuclei, that is, one is haploid and the other is triploid. Thank you.